Hello and welcome to the point of Hussein Biology. Today we are going to discuss the very important topic, and that is about an instrument called microscope. So how microscope was discovered and when it was discovered? Initially microscope was designed by Zacharias Janssen, and further it was modified with time and now we have the original form of microscope. There are two types of microscope: compound microscope and electron microscope. So what is compound microscope and what is electron microscope? Compound microscope just use light for the magnification and resolution of sample, and electron microscope use electron beam for magnification, and exactly the electron microscope will make the 3D image while the compound microscope will make the 2D image. Okay, so that is the difference between compound and electron microscope, that compound microscope uses light and electron microscope use electron beam. Okay, so we have the compound microscope, it is also called light microscope. So have a look at how we will use the compound microscope. It's quite easy, you will follow me step by step. First of all, you should have to know that what are the different parts of this microscope. So have a look on this. Yes, have a look on this one. We have here is the light source. Okay, this is the light source, and we have the fine focusing. Okay, the fine focusing and the coarse focusing knobs. Check. And yes, we have the eyepieces, and we have the special 4 objective lenses. Okay, the lenses are 4x, 10x and 100x and 40x. Okay, so there are different uses of these lenses. These are called objective lenses, and this is the stage where we put the sample. Okay, and this is the arm, this is the body, this is the nose piece. It's okay, these are the ocular lens, also called eyepieces. So these are the parts of microscope, compound microscope. Okay, so how we will work on this microscope? So it's easy. Have a look on this. We need some things very importantly. This is a slide. This is a slide where I have a slide box. Okay, it's quite easy, you can take it from the market. This is a slide box, and also we have the box of core slip. Okay, it contains core slip. Yes, the very small glass, square glass. Okay, so these are used and during when you are examining something. So if you are magnifying something on microscope, so you should need these things. Okay, so for example, if you wish to take an overview on the urine sample, so how it will be possible? First of all, you should have to make a sediment. How the sediment is possible on the centrifuge. You should take the sample and a tube, and you should centrifuge that urine for 5 minutes. Okay, and then you should take the sediment on a slide. For example, this is the slide, so you should take the sediment. Okay, the last the sediment which is the remaining, and the part which sediment at the bottom of the tube. Okay, so you should have to take that part, that is called sediment, and you should have to take a drop of that sediment on this slide, and then you should have to take the core slip. Exactly, I will take a core slip, and for example, if I put down the urine sample sediment here on the slide, then I will put this core slip on this. Okay, so now what I should have to do? I have to do one thing, that first of all, there are two things which are very important in microscopy. First of all, what is focusing, and another is coarse focusing. Okay, so initially you should have to do coarse focusing. Okay, then you should have to do fine focusing. Okay, just for urine focusing, you should have to use 10 lens. Okay, if you focus the urine on the 10 lens, then you will properly magnify and do resolution and contrast and magnification properly at 40 lens. Okay, so that's all about the urine. Okay, now if we have to study the blood smear, so how we should have to take a drop of blood on the slide. Okay, we should have to take a drop of blood on the slide. I have a make a Yes, uh, before 10 minutes I make this smear. Okay, so you should have to make a finger like smear. Okay, then you will dry it, then you will put the methanol on it in order to fix it, and then you will put the ginger stain. Okay, any of the rum and whiskey stain on it. Okay, after staining, then you will properly examine, observe it on the oil emergent 100 lens. Okay, so during examination on the 100 lens, you should have to take focus on it on 100, and you should have to set the light high. Okay, so it's all about the microscope. And I think urine examination and blood smear examination is very important. Okay, same as for stool examination. If you are going to do stool routine examination, so same like that, you should have to take the stool okay, in the normal saline. You should have to centrifuge it properly, and then you should have to take the sediment from it on a slide, and you should cover with it with the cover slip, and after that you should examine it on 40. Okay, so it's all about the microscope, how to use it, and it's quite easy. I think it will be the more beneficial lecture for you. Yes, exactly, I will come with the treatment of coronavirus exactly after a week. So subscribe my channel, like it, and share it with your friends. I will come with a very important lecture in the next. Thank you.